and I'm going to be presenting my final presentation for statistical analysis of genetic data analysis project, final paper presentation, and some background information. In a broad sense, what we sought to do was compare and contrast the findings of statistical analysis done using SAS-based product called JMP Genomics 9.1 with the findings of an interval mapping analysis performed using R with QTL Package. And the source article we used has been presented already, but I'm going to just have to show it again for background. But before that, I want to go over our goals. And the goals of our group project were to analyze the data using R with the QTL package, but without covariates like our source article. And the source article we used had covariates like hyperlipidemia, cholesterol, and more. And we also wanted to analyze the data using JMP Genomics 9.1, which is available through UF repositories. And we wanted to compare both of our group analyses with the analysis from our source article and see what we got. So for background information, this has already been gone over by Sharvy and Paul. I just wanted to note that I added this aortic root arrow here because this is the aortic root. I don't think it was labeled well before. And this is where the aortic artery and the heart meet each other. Okay, so the background, the data set is from the JAX laboratory, which is an independent nonprofit biomedical research institution, and they provide these genetically modified strains of mice for research. And the original paper is in the National Institutes of Health repositories right here. And so what I want to talk about, especially with my background as a dietitian, is atherosclerotic heart disease. And according to the CDC, heart disease is the leading cause of death in the United States. And the CDC estimates about 650,000 Americans die each year from heart disease alone. And this is my source here, cdc.gov, heart disease facts. And one of the main types of heart disease is atherosclerotic heart disease. And atherosclerosis is the deposition of plaques of fatty material on the inner walls of the arteries of the cardiovascular system. So what is the aorta? The aorta is the largest artery in the body, and over the course of many years, this plaque buildup causes a narrowing of arteries and makes it more difficult for oxygen and nutrients to be transported from the heart to the rest of the body. And it begins at the top of the left ventricle of the heart, pumping oxygen-rich blood through the aortic valve and into the aortic arch. And there were 5 phenotypes measured in the study. Each phenotype is the measurement of atherosclerotic plaque in 5 separate locations of the aorta. And these 5 locations are the the root of the aorta, the inner curve of the aortic arch, the innominate artery, the left common carotid artery, and the subclavian artery plus its upper wall. So now what is apolipoprotein E? And apolipoprotein E is known for playing a role in both cardiovascular disease and even Alzheimer's disease as well. And our study focused on the role of apolipoprotein E gene, which is commonly referred to just as ApoE. And ApoE provides the instructions for making the protein called apolipoprotein E in cells. Without the apolipoprotein E gene, apolipoprotein E cannot be made within cells, and the lack of apolipoprotein E makes the body highly susceptible to hyperlipidemia, hypercholesterolemia, atherosclerosis, and therefore cardiovascular disease, which ultimately can lead to heart attack and stroke. And this makes studying animals like mice that are bred to be ApoE deficient or ApoE null crucial to our knowledge and development of drugs to better treat cardiovascular disease in humans. So some of the genetic background here, the ApoE gene has at least 3 alleles, which are ApoE2, ApoE3, and ApoE4. The most common allele in the general population is the ApoE3 allele, which is found in about half of all humans. And our data set was created by using the F2 progeny from a cross of ApoE-lacking mice strains, which are the E null mice. And the F2 progeny mice were created by crossing enal mice on the DBA ApoE marker, location, and the B6 ApoE genetic backgrounds bought from Jackson Laboratory. And so this is done by creating first male DBA ApoE mice, mating them with female B6 ApoE mice, and generating their F1 progeny. And those F1 hybrids are then intercrossed in order to create the F2 progeny, which were used in the study. And the F2 progeny were compared with two previous F2 crosses using Apolipoprotein E null mice on the 129S6-SVEV TAC or 129-ApoE location and DBA ApoE location backgrounds, along with the B6 ApoE and 129 ApoE backgrounds. And each parental strain of mice was crossed with two others in what is called round-robin crosses. And with round-robin crosses, the round-robin crosses are done because allele frequency calculations for inbred populations can be very challenging. And calculating these frequencies on non-inbred corrected data can be biased due to samples that are not independent. So allele frequencies that are calculated on inbred corrected data can bring about unnatural increases in the significance of rare alleles. Therefore, the allele frequencies can be calculated using a method called round-robin, which can minimize both of the previously listed unwanted effects. And the round-robin method has been laid out in detail in a source listed as Parks and Worth, 1993, and that paragraph was summarized from an R blog using the following link, which is in the final report. So in the source article, 136 female and 96 female F2 mice were aged 4.5 months, euthanized, and then dissected to examine the atherosclerotic plaques in the inner curve of the aortic arch and the aortic root. And the cross-directional notation of the two F1 strains used to create the F2 progeny is B6 ApoE times DB ApoE F1 times B6 ApoE times DB ApoE F1. And the methods that we used using JMP Genomics 9.1, which Paul Dean described in his report and presentation. And this analysis is done using JMP Genomics, which uses a graphical user interface, so there is no source code. And the JMP Genomics 9.1 created relatedness measures, relationship matrices. It created comparisons between genomic-wide association testing with the single nucleotide polymorphism trait association, along with linkage maps and the quantitative trait loci single markers, along with linkage maps comparisons, along with quantitative trait loci and composite interval mapping and other types of mapping, which I won't go into. So the methods using the R QTL package that we use. They are using single marker QTL analysis. Analyzing multiple phenotypes with each phenotype considered individually. The QTL analysis was done using the EM algorithm, using the scan1 function to perform single marker QTL scans, and the significance LOD or log odd score thresholds were obtained using 1,000 permutations with a significance value of 0.05. And I wanted to show here was the Manhattan plots that Paul used. I happen to like them because they happen to show the LOD peaks here. For example, at chromosome 2 here, chromosome 2 again here, chromosome 2 here again, and chromosome 2 here again. So we can see that chromosome 2 plays a role in almost 4 out of the 5 phenotypes in the JMP Genomics study. And here, the QTL significant single markers by chromosome, and we can see chromosome 1, 2 here, chromosome 2, 6, 10, 11 here, chromosome 2, 3, 6, and to a lesser extent 10 here. And in chromosome 1 and 2 here, chromosome 8 here, and then here finally in phenotype 5, chromosome 2, and Paul stated these final conclusions here, so I won't repeat them. And so for the R analysis that we did using our QTL package, and this is the genetic linkage map, and you can see here that chromosomes 1, 2, and 10 play a large amount of role in the genome-wide map. And here in the R package, we went to the LOD curves, and we can see here, just like in JMP Genomics, for the aortic root here, we can see that 1 and 2 plays a role, along with some other chromosomes, and the phenotype 1, which is the inner curve of the aortic arch, the highest LOD or log odd score observed was for chromosome 2, and also in chromosome 10, which was similar to our source article. Keeping going here in phenotype number 3 with square area 2, which is the innominate artery, and also here the highest LOD peak is chromosome 2. And here again with phenotype 4, we see chromosome 1 and chromosome 2 playing a large role. And in the final phenotype 5, we see chromosome 2 again playing a large role. So the conclusions from our source article, I wanted to discuss quickly that JMP Genomics version 8.0 was actually used to analyze the trait data in the source article. And one-way analysis of variance was used to compare multiple groups in trait values and gene expression amounts in the aorta. Then they used Tukey-Kramer HSE test in JMP Genomics 8.0 along with Shapiro-Wilk and Kolmogorov to analyze the normality of the F2 mice. And square root transformation of values was needed for each phenotype for all aortic locations in all of our studies, for all 3 different studies, from the article, from our study, from the genome study using JMP 9.1, and then in the source article using 8.0, all had to be square root transformed because the distance was found in micrometers. So that square root transformation did help for the data to become approximately normal. And the conclusions from our source article continued here. One of the main findings of our source article's analysis was that the B6 times DBA ApoE-F2 population, the discovery was that the significant QTL was on chromosome 2 plus a suggestive QTL on chromosome 10, which does impact plaque lesions in the aortic arch, which is phenotype 2. And then to sum up, finally, we're already at 13 minutes here, so the final conclusions, a discussion of results, which are in the final written report in further detail. All 3 analyses found similar results, which were that chromosome 2 in all 5 phenotypes using RQTL with or without covariates, as well as with JMP Genomics 9.1, along with the supporting role of chromosome 10. And the chromosome 2 appears to play a large role in all 5 phenotypes, all 5 locations of atherosclerotic plaque within the aortic valve, the aortic artery. And finally, the final and most important point I want to drive home is that the SAS product, JMP Genomics 9.1, just has an easier to use graphical user interface, which means there's no source code, and it's more alive and more entertaining graphics, for example, the Manhattan plots and the other types of plots that Paul showed. And again, in my analysis, just showed that if you're having to entertain non-statistical audiences, that it's a much more alive and efficient way to entertain non-statistical audiences. And I want to thank you very much. My name is Christopher Powell, again, for our group with Sharvi Bihat, Paul Dean, and myself, Christopher Powell. And thank you.